Hello, my name is Annabel Cho, and I am a product manager with Meissner. Today, I will review Meissner's advances in barrier filter technology for improved performance and reliability in aseptic processing where PUPSIT is deployed. Meissner has worked to improve performance and reliability within the barrier filter technology space. Zebragard features a uniquely patterned PVDF membrane, which, coupled with our pleated membrane technology, affords the industry's fastest airflow recovery after filter wetting while maintaining exceptional air and water flow rates. Let's take a closer look into Zebragard's membrane and what makes it so special. Zebragard features a uniquely patterned PVDF membrane with hydrophobic and hydrophilic portions within a singular membrane, and coupled with our pleated membrane technology, affords the industry's fastest airflow recovery after filter wetting. This PVDF membrane meets the ASTM bacterial validation standards and is compatible with both gamma and autoclave sterilization. Additionally, it implements seamlessly in single-use systems through customization of inlets, outlets, and vent filtration. The use of sterilizing-grade filters for aseptic processing is a highly utilized method for its reliability and assurance. Yet, filter integrity must be routinely confirmed via non-destructive tests such as bubble point, diffusive flow, or pressure decay. Although mandatory integrity tests are typically performed post-use, recent regulatory guidelines have solidified the enforcement of pre-use testing. In particular, the newest revision of the European Union Guidelines to Good Manufacturing Practices, Annex 1, now explicitly calls for pre-use post-sterilization integrity testing, otherwise known as PUPSIT, where barrier filter technology is typically deployed. PUPSIT was brought forth to address concerns of filter flaws created from filter manufacturing, transport, installation, and sterilization, which would allow passage of contaminants downstream, compromising product sterility and patient safety. These theoretical flaws would clog during filtration and result in a past integrity test post-use, otherwise known as filter flaw masking. Although PUPSIT helps address filter flaw masking, opposition exists for the added complexity of PUPSIT systems to allow for inline filter wetting and drying without breaching system sterility. This is where barrier filters really shine. Where integrity testing a sterilizing-grade filter inline, care must be taken to evacuate wetting fluid from a system without breaching sterility, and proper system ventilation must be provided to avoid pressurizing your system or receiving inaccurate integrity test results. Although differing methods of sterile boundaries exist, by placing a barrier filter downstream of a product filter, system simplicity is preserved and additional flexibility is unlocked. Alternate methods, such as a catch can or a flush bag, limit the volume of wetting fluid which can pass through a system, limiting your ability to re-wet and retest a sterilizing-grade filter if needed. System drying also becomes increasingly difficult due to pressure concerns. Similarly, the use of a filter arrangement which requires two filters, one hydrophobic and one hydrophilic, increases system complexity, which introduces risk. The use of two filter elements requires manipulation of the system during testing and requires testing the integrity of an additional filter prior to product filtration. Meissner has worked to improve performance and reliability within the barrier filter technology space. Zebragard features a uniquely patterned PVDF membrane, which, coupled with our pleated membrane technology, affords the industry's fastest airflow recovery rate after filter wetting while maintaining exceptional air and water flow rates. Here we can see Zebragard in action through a graphic of a sample PUPSIT flow diagram, where if we follow the flow of wetting fluid through the system, we can appreciate the evacuation of the fluid through a barrier filter. Similarly, by looking at the flow of air through the system, we can appreciate air escaping through a barrier filter. There is great benefit there, since filter integrity test failures commonly occur due to improper filter wetting, and Zebragard offers unlimited volumes of particle-free water and air to be flowed through the system. Next, we will look into airflow recovery. What is airflow recovery and why is it important? Airflow recovery refers to a barrier filter's ability to recover its typical airflow rate if the filter were dry after it is wet. What happens when wetting a barrier filter is that water beads will form on top of the hydrophobic portion of a membrane from water being repelled, which effectively blocks the passage of air and may create back pressure. It is important to quickly dry the hydrophobic membrane to ensure integrity test values are accurate and there is no hindrance created within a system. Additionally, rapid airflow recovery leads to reduced system drying times for a decrease in process time without product dilution. Zebragard's performance and reliability can be appreciated in the graphic, demonstrating consistency in airflow recovery and industry drying time. This can be further analyzed through a different chart, where we can appreciate Zebragard's outstanding airflow recovery times, coming in at under 5 minutes at 2 differing pressures. I hope you enjoyed learning more about Meissner's Zebragard barrier filters for final fill and finish applications. This device delivers all of the benefits of a hydrophilic and hydrophobic PVDF membrane filter into one compact design that seamlessly integrates into manufacturing operations where PUPSIT is deployed. For more information, please visit meissner.com/Zebragard.
Hi, thank you for joining us today. My name is Thomas Lazzara, and I'm Director of Research and Development at Meissner. We'd like to talk about an issue that has been raised by users of tangential flow filtration products. Specifically, that they experience variability in the hollow fiber filters they receive, which means that they have to compensate by modifying process parameters. If this sounds like an issue you're experiencing, then please join me. I will share with you how our filters can benefit your manufacturing operations. Today, I will highlight how improved membrane characterization leads to better process stability and repeatability in regards to our hollow fiber product line, SepraPor. The membranes we will be discussing today are specifically in the form factor of hollow fibers. As most of you may be familiar with, these hollow fibers are operated in a tangential flow filtration mode, with the bioprocessing fluid flowing parallel to the membrane surface with a sweeping action that reduces membrane fouling. Today, we will cover 3 discussion points for these membranes. First off, a brief product introduction, followed by membrane characterization and product consistency, and finally, a demonstrated application involving the purification of plasmid DNA. SepraPor filters are constructed with high-performance polymers and proprietary thermoset resins, and are designed for the most demanding applications. Listed here are the materials of construction for these filters. The membrane housing, ports, and connectors are all made of polysulfone polymer, while the netting is made of polypropylene, and the fiber encapsulation is achieved with epoxy. We offer our hollow fiber filters in effective filtration areas from 10 square centimeters to 8.3 square meters, which is sufficient to cover the full range of your different processes, for process scalability and flexibility. From small-scale benchtop reactors operating at much less than 1 liter to full-scale bioreactors operating at hundreds to thousands of liters, our solutions offer process solutions and performance at every scale. Currently, we offer microporous filters with 0.1, 0.2, and 0.45 micron porosity ratings. Our ultrafiltration filter line has retention ratings of 10 to 750 kilodalton. Shown here is a major difference between the actual pore size in nanometers of the different porosity denominations of various types of membrane, from ultrafiltration to microfiltration. Traditionally, microporous hollow fibers with larger sub-micron pores are used in upstream processing, where larger cells are part of the bioprocess, while ultrafiltration is typically used downstream, where proteins and smaller constituents are the targets to be purified. More recently, with a variety of new therapeutic manufacturing paradigms, both microfiltration and ultrafiltration have been used as separation and purification elements in many recent fields, such as mRNA, VLP, and emergent cell and gene therapies involving viral vectors and lipid-based vectors. Illustrated on this slide are different therapeutics and expression vessels used in various bioprocesses, where we highlight the importance of selecting the proper pore size in relation to the size of the bioprocessing element. This is exactly why we believe in providing our customers with the best characterized and most consistent membranes on the market to help them achieve repeatable performance over time. In this next section, let's discuss the particular aspects that differentiate our hollow fiber membranes and provide them with a superior quality than our competition. We have worked extensively on membrane characterization, as we believe it is of the utmost importance for our customers to receive products that are both highly performant and consistent. We hope that you will agree that well-characterized membranes allow your processes to run at peak performance consistently. Improving characterization leads to better membranes, with accurate and precise porosities and better lot-to-lot consistency. First, let's look at the membrane structure we developed. All our hollow fibers membranes have low asymmetry and have a dense structure. We developed this dense structure without compromising the flux. This is shown on both the schematic here and the microscopy picture to the right. This almost isoporous membrane has these advantages. It's robust, concentric, and dense, with high flow rates, no breakage or defects during operation, better performance under high and variable pressure differentials, and is resilient to repeated uses and steam sterilizations. Microfiltration and ultrafiltration membranes differ in their pore sizes. Their characterization is also different, and the methods used depend on the pore size rating. For microporous membranes, bubble point and bacterial retention tests are typically carried out to provide porosity values. For ultrafiltration membranes, their porosity is typically characterized by secondary methods involving molecular weight markers that may be proteins or polymers to obtain a molecular weight cutoff, MWCO. The MWCO is obtained from the passage of molecular weight markers using the filtration test and scientific instrumentation to determine the degree of membrane crossover. For microporous hollow fibers, the porosity is determined with bacterial retention studies, similar to how other bioprocessing membranes are characterized. We have developed process control strategies that allow us to keep narrow specification ranges for our membrane porosities. In comparison with a competitor, we can see that the specification ranges are 2 to 3 times more narrow. When product specification ranges increase, we also expand the variability in both pore size and fluxes. The reduced variability in our product specifications results in improved lot-to-lot consistency and lower process variability for our customers. Another way to assess the reproducibility of our membranes is to test both the membrane resistance and assessing the pore size distribution of different membrane lots manufactured. Membrane resistance is a measure of pore size and pore size distribution, where higher porosities have lower membrane resistance. As seen, this is indeed the case, and different lot tested tend to cluster around distinct averages. In addition, looking at the pore size distribution of different lots shows that our manufacturing process produces reproducible membrane structures. Therefore, these characterization efforts demonstrate that indeed our manufacturing process produces lot-to-lot reproducibility, which is important to have reproducible membrane structure and permeability and reproducible membrane pore size distribution. Now in this second part, we present a quick overview of how ultrafiltration membranes are characterized, because they are defined by the molecular weight cutoff rather than microns like microporous membranes. We use a minimum of 3 molecular weight markers with known molecular weights to challenge the membrane, 1, 2, and 3 being small, medium, and large polymers of different molecular weights. To determine the molecular weight cutoff of the membrane is challenged using these markers, and the percent passage through the membrane is recorded. The percent passage is illustrated here through the red arrows. Depending on the percent passage at different molecular weight markers, we can assign a molecular weight in kilodaltons. The advantage of using 3 molecular weight markers becomes apparent when comparing multiple porosities. For 2 different molecular weight cutoffs, say 100 and 300 kilodalton membranes, the passage ranges at different molecular weight markers have overall lower overlap, shown on the left graph for SepraPor. Supplier A, however, has 100 kilodalton range determined using the same molecular weight marker as the 300 kilodalton membrane, and there is significant overlap of the passage ranges. You therefore have product specification overlap. If you end up having product specification overlap, then you automatically increase your product variability. Taken together, these passage ranges for different molecular weight markers generate what we call passage bands. Shown here are 3 lots of membrane that are classified as 300 kilodalton, showing that they fall within the appropriate passage band and fall within the acceptable range for each molecular weight marker. What differentiates SepraPor ultrafiltration hollow fibers is identified in this table. We use 3 molecular weight markers, not just 1. We have a distinct retention behavior described by a passage band. We therefore have distinctly defined molecular weight cutoffs, and we are confident that this provides better consistency. These passage bands are unique to each molecular weight cutoff, with minimal overlap between them. Here is shown 10 kilodalton to 750 kilodalton passage bands. SepraPor therefore provides a more in-depth membrane characterization than other membranes on the market because it has defined and unique passage bands for ultrafiltration membranes. So we realize this is a more demanding characterization, but we are confident that it improves lot-to-lot consistency and will translate in low process variability for our customers. Just to wrap up the membrane data we discussed today, we would like to emphasize that better characterized membranes lead to reduced process variance and improved process stability. Accurate and precise pore size assignment, together with a robust membrane structure and reliable high fluxes, are particularly useful for customers as they allow more predictable lot-to-lot filtration and therefore reduced long-term process variability. So in this final section, we would like to highlight a recent SepraPor ultrafiltration application, where it was used in the purification of plasmid DNA, specifically in UFDF steps. In this process, shown in the diagram, SepraPor was used in 2 UFDF steps prior to chromatography and prior to sterile filtration. Using SepraPor, we demonstrated that we can achieve process intensification in comparison to traditionally used cassettes. Ultimately, hollow fibers allowed us to overcome design limitations in cassettes that prevent high filter throughput, as illustrated in this graph. In cassettes, inlet pressures spike when trying to increase the feed flow rate while trying to gain efficiency. In comparisons, hollow fibers allow much higher throughput rates, therefore more effective filtration. This table highlights the performance difference between cassettes and a SepraPor hollow fiber. We can see that using SepraPor with much lower surface area operated at double the flow rate of a cassette allowed to reduce the amount of diafiltration volumes and reduce the processing time below 5 hours in comparison to 8 hours. Overall, we saw that using SepraPor allowed to reduce the filter surface area by over 50%, allowed greater than 25% savings in buffer consumables, and reduced the overall processing time by over 40%. In conclusion, we have demonstrated today how SepraPor has been developed with higher industry standards to improve characterization, which translates to accurate and precise porosities and improved lot-to-lot consistency. Now, if you haven't taken any notes, please remember this takeaway. Well-characterized membranes allow your processes to run at peak performance consistently. So I'd like to thank you for your attention today. And if you do have TFF needs for your demanding applications, please don't hesitate to contact us. You can visit our website at www.meissner.com/SepraPor. We also have resources available online, which include videos that you can consult and additional documentation. And again, if you do have questions, please reach out and we'll be happy to answer and help you in your TFF applications.